Well, good morning. I am talking about microbial ecology and marine conservation. I work at the Metropolitan Autonomous University in near of Mexico City in the environmental science department, and I work with microbial communities. All my work in my research are about microbial communities. Microbial communities in probiotics, in adults, microbial communities in mice underwent stress, microbial communities in the antimicrobial resistance and pathogenicity in municipal wastewater treatment plant, wherever. So all my work it's about microbial communities. I have 2 years ago the effect of the probiotic Lactococcus lactis in a freshwater prawn, Macrobrachium rosenbergi. So all my work are in microbial communities. So one thing that I been working in my lab, it's the use of bioinformatics. So the bioinformatics now it's a very important issue because with the new technologies of massive sequencing platforms, we have to analyze a lot of a huge amount of information. We use metagenomics approach. We use a metataxonomic approach. We are using a lot of molecules that we can study to make a microbial ecology, to make a function ecology. So we are using bioinformatics. So one of the huge things that my lab does is the use of the new computers, something like this. We have one in the campus at the university that called Nautilus. That is a huge computer that can process a lot of things, that can process a lot of sequences. So we are working with this. So one thing that my students have to learn when stay with my group is the bioinformatics. So the other thing that is important to know is in the actually is the new sequence platforms like this. This is a new platform to sequence in the field. You can use here in the boat. You can use in the field, in the forest, wherever you are working. You can use this kind of platform. These platforms works with DNA and works with RNA, and it's possible to use in field. It's possible to use in the lab, but the important thing is that it's cheap now. You can use one platform of this about $1,000. So they generates a huge amount of information. So one thing that we are doing in our lab is using this kind of platforms to obtain a huge amount of information. So what the corals? What is our work in the corals? We are working in microbial communities associated with healthy Montrastea cavernosa coral and with a stony coral tissue loss disease. This is a disease that appears probably in 2018 in Florida, and we are found in 2019 in Mexico, in the Caribbean, in the Mexican Caribbean, and it's a disease very dramatic because all the corals die about 1 week, 2 weeks, no more. So what are the coral reefs? They are one of the most important marine ecosystems, as you know. We have a huge amount of environmental health variables like temperature, acidification, nutrients, competition. So 25% of the productivity in these ecosystems are associated with marine species. And we have found loss of homeostatic state, emerging diseases, invasive species, environmental contamination, and we have found an environmental sensitivity to associated with all these systems. The coral reef represents only 0.2% of the seafloor cover, but wherever is one of the most marine ecosystems in the most vulnerability to the climate change and the anthropogenic impacts. The geographical position makes them vulnerable to multi-directional impacts, and Montrastea cavernosa distribution is in the Caribbean and Atlantic Ocean, considered an extreme generalist. Our first approach is to find which is the responsible of this disease. It's a bacteria, it's a virus, it's a what, okay? This is our first approach. So we have global climate change, increase in the sea surface water temperature. This provokes coral bleaching, emergency diseases, frequency and magnitude of natural phenomena. In marine water acidification, we found higher rates of calcification, decrease in the reproductive or immune metabolism, and the average sea level rise. We found coastal erosion, change in environmental quality. So the stony coral tissue loss disease is a disease yet unknown causative agents in 2014 in Florida, sorry, not 2018. They observed multiple lesions whose sign of the presence of white tissue circular areas, and this disease is transmitted by direct contact or through the water column. So we can find some this in Mexico, in the Tulum area, in Playa del Carmen, Cozumel, and Chinchorro. So and now we believe that even in the north of the Mexico, we found this disease. So in the Mexican Caribbean, the occurrence was detected in 2018. The affectations reached approximately 20 species. It's detected in all regions and marine natural protected areas in Quintana Roo. And we leading to the functional extinction of last one species, the Dendrogyra cylindrus. That probably is extinction of this species of coral. It's important to concentrate research efforts in different areas, including molecular pathology, where studies or changes in the microbiome in the disease process are carried out. So we are working in try to answer this kind. So one concept that we use to study all these diseases in the corals is the concept of the alluviont. For us, the alluviont is very important because probably when I start, I'm not biology, I am biologist, I am a biochemistry engineer. So when we start to work in this kind of things, we use this concept because we are not alone, okay? Probably if you remember the teachings in our child, they said this is a plant, this is an animal, this is a bacteria, this is a eukaryote or a prokaryote cell. But we think, because it's now a truth, that we established a lot of interactions with another cells. For example, a coral here can establish interactions with zooxanthella, can establish some interaction with virus, can establish some interactions with bacteria protozoa and with fungi, okay? So one thing that we are doing in our lab, in my lab, it's to study this kind of interactions at the molecular level, okay? So we are studying DNA, we are studying RNA, and we are trying to answer what kind of interactions are present in the coral, or between the virus and the bacteria and the fungi, or the zooxanthella, the algae. So the ecological interactions of alluviont, the interactions of the coral alluviont are extremely important for maintaining the health of the animal. Disturbances due to the chemical, physical, or mechanical impacts are reflected in the distribution of the microbial community, making it vulnerable. So one thing important for us is the homeostatic state of the alluviont, how to measure these kind of interactions, and high strain levels, loss of the homeostatic state, disruption of ecological interactions. So what we are doing? So we are we are measuring. We have these sample sites in, this is Playa del Carmen, this is Tulum, and this is Cozumel. And we are taking some samples of the mucus of the corals and making some metataxonomic approach. The metataxonomic approach uses this marker, the 16 rRNA marker, and this can measure all the bacteria that are associated with the corals. This is the different species that we were working: Porites astreoides, Montrastrea cavernosa, Dendrogyra cylindrus, Agaricia agarites, Siderastrea, and Porites furcata. There's some of the corals that we were working in 2019. So the results that we found showed a high diversity of bacteria in both diseased and healthy corals, because we have a group of healthy corals and a group of bleached corals. So the total taxonomic diversity of bacteria was 174 generally distributed in 27 families. We are working with this. This is one of the results, the PCA plot, that is a multivariable two-dimension plot that measures. We have the blue ones has the disease, and the yellow has healthy. So we have the distribution of the microbial communities. Every dot or every figure in the plot is a microbial community. It's not only a coral, it's not only a population, it's a real microbial community. So we can see some difference. This is the healthy. The 3 points are the healthy, and the distribution of the sick is down and different. So we studied this kind of plots. This is a corona plot. In this corona plot, we are making some function bacteria associated with the corals. We can see some protobacteria. We can see some bacilli, firmicutes. This is Oceanospirales. This is the picture of the microbial community associated with these corals. So one example of all the work, because I don't want to sleep now, to sleep, is Rhodobacteracea. According to Roder, this bacterial group is opportunistic, where there is an immunological vulnerability highly, that their dominance in diseased corals is remarkable. 72% within an alpha protobacteria or Rhodobacteracea, well, diseased corals have a dominance of 99%. For us, this is important because one strategy to control this group, this bacterial group in the disease, the first that we think is to use an antibiotic. But if you use an antibiotic in the ecosystem, you probably can cause a bigger problem, because the antibiotic works not only with Rhodobacteracea, it works with another species. So now we are working with a specific target that is a virus that kills Rhodobacteracea. The advantage of using a virus is the virus recognizes a specific sequence of the Rhodobacteracea genome and kills the Rhodobacteracea. This is, for us, this is an advantage because we can apply these viruses in the specific form. So we don't cause a bad impact because we are using a specific target of these kind of things. So another thing that we found in these microbial communities is the presence of Vibrio fischeri is another bacteria, sorry. But this bacteria has a special process that called quorum sensing. The quorum sensing is a communication system in bacteria, and this communication system produces light. It's a bioluminescence cluster of genes that produce light. If you see Lux M, Lux N, Lux Q, Lux P, Lux, all the genes that call Lux are genes that are associated with the light production. For us, the light production again is we have two. I have two students working with this because the light, the light in these corals we can use this light to mark all the diseased coral, not only the bleach. They can produce this light, and we can found this light with a special lamp. So all the past pages are about the work that we are doing, and what we are doing now. We are in the current research proposal. Our group starts to find the bad guy. Okay, so now we are looking the resistance. Who is the microbial populations that could improve resistance in the corals? Because when we are diving in the Caribbean, we found some corals that they don't have disease. So for us, the first thing that we think is genetic. But when we compare the genome of one coral, the healthy, and the coral of the sick, the genome is the same. So the next thing that we think is, if we don't found difference between the genomes, probably the difference is in the microbial communities associated with the corals. So we are working today in found which components of the microbial communities can reach resistance to the corals of different diseases, principally the coral disease, the white coral disease. So through the metagenomic analysis of disease and healthy Montrastea cavernosa tissue and of the water surrounding the corals, the microbial community will be characterized metataxonomically and metafunctionally. Water quality will be evaluated to identify possible abiotic stressors that may be related to the disease. The difference between reefs not records of the sickness compared with the site with the disease will provide data for coastal ecosystem management. So we are working with this. So we have different sample sites again in Playa del Carmen and Cozumel, and we are working in this with a metagenomic approach. So finally, we don't have results in this part because the project starts 2 months ago. So we are only in the sample sites and these kind of things. But we can collaborate with you in these lines. Microbial studies in diseases. I talk with someone of you that we can collaborate in this study. We can collaborate in disturbed and non disturbed ecosystems. We can collaborate with you in the Holobiont in measuring the interactome at the DNA level. For us, in the microbial communities, one special thing is to work with the genetic markers, because when we are using these special platforms to sequence all the material genetic material, we can establish who are in the samples, which is the population that are present in the samples. But even we are using this approach to study which kind of genes are codified in these populations or in these communities. We can find genes associated with energy, genes associated with metabolism, genes associated with wherever you want. So we can cross and make some interaction net between genes in these communities. And finally, we can do functional ecology about these ecosystems. So this is my group, and thanks.